Hello, and thank you for attending this presentation on enabling biomarker discovery and screening with Fireplex multiplexed assays. My name is Emma Ghaffari, and I am the Field Application Scientist for our multiplex capabilities, and I am responsible for providing technical support for our customers in using our platform here at Abcam. The drug discovery pipeline lasts on average 12 years and can cost up to $1 billion. The early stages of drug discovery have particularly high failure rates, with only 5% of compounds making it out of the target identification to lead generation stage. There are several factors that can contribute to this failure, including issues with pharmacokinetics and pharmacodynamics, as well as efficacy and safety issues with lead compounds. Safety and efficacy issues represent, in fact, two of the most common causes of compound attrition, and that is not only true for the early discovery stage, but also for the clinical development stage. Therefore, developing tools and assays that can assess early in the process whether a compound can have its targeted effect and be safe has the potential to vastly improve the early stages of the drug discovery pipeline and increase the number of drugs that can make it to market. Proteins have traditionally been used as ideal biomarkers as they are readily available in circulating biofluids, such as blood, plasma, and serum. Protein biomarkers are used to detect changes in prognosis, diagnosis, and different disease states. The changes in gene regulation and protein expression have been highly indicative for disease progression and our understanding for the drug discovery pipeline. Protein analytes are, however, susceptible to sample heterogeneity in how these samples are stored. Freeze-thaw cycles can affect the stability of the proteins in these samples. In the last decade, microRNAs have become emerging biomarkers. They are found in normal circulation and they are more stable compared to their protein counterparts and are not susceptible to degradation when going through freeze-thaw cycles, thus enabling the possibility to review whole body health and toxicity directly from biofluids. The last decade of research has revealed the large potential for using circulating microRNA signatures in the detection and treatment of cancer. These efforts have been supported by the finding that microRNAs have tissue-specific expression patterns, making it easier to link changes in circulating microRNA levels to organ or site-specific diseases. Altered microRNA expression has been found to correlate with cancer stage and progression. Several studies have identified a role for microRNAs in epithelial to mesenchymal transition, further pointing to a potential role for deregulated microRNA expression in tumor metastasis. Therefore, how can we develop reproducible multi-analyte signatures to assess disease incidence when working with limited volumes of precious samples? So in that context, I would like to introduce you to the Fireplex platform, which was specifically designed to aid in the areas of biomarker discovery and validation, with the idea to enhance the overall efficiency of the drug discovery process. The Fireplex technology from Abcam was developed at MIT nearly 10 years ago and uses optical liquid stamping of hydrogel particles to create a platform for discovery and screening of both microRNA and protein biomarkers. For biomarker discovery studies, Fireplex offers multiplex assays that are highly sensitive, run in a 96-well plate format, and are read out on standard flow cytometers. For biomarker screening studies, Fireplex offers multiplex assays that are rapid, have automation-friendly workflows, and are run in a 384-well plate format, and are read out on high-content images. The Fireplex technology is based on a sandwich immunoassay principle, where each particle code acts as an individual well, allowing for a highly multiplex assay format. Fireplex uses fluorescent dyes for both the particle barcoding and analyte quantitation. Particles contain 3 functional regions, indicated in color, and that are separated by 2 spacer regions. In the 2 coding regions on either end of the particle, different levels of yellow and green fluorescent dyes are used to create a maximum of 70 unique coding combinations, allowing analysis of up to 70 analytes in each particle mix. The central region of the particles contain the analyte quantitation region, where capture antibodies are used to detect the protein analyte of interest. Here, a red reporter dye is used to detect the captured analytes. The kit contains protein standards which allows for absolute quantitation of analyte binding if required. Data analysis is conducted using our free, fully integrated bioinformatics software package called Fireplex Analysis Workbench, which can be downloaded from our website. Our software utilizes the raw data generated by the flow cytometer or high content imager to do particle decoding and analyte quantitation. A range of analysis can be performed in the software and exported in a CSV format, which can be further analyzed in other software packages like Excel or Prism, or any other alternative software package that the researcher uses. Abcam is known for its extensive range of high-quality antibodies used in a variety of applications. Using our rabbit recombinant monoclonal antibody technology, it allows us to generate high-quality antibodies that are consistent over different batches, which is critical for screening applications. We extended this antibody portfolio to generate antibody pairs using the same recombinant technology. These pairs are used for a range of assays, from our matched antibody pairs kits to our simple step ELISA kits. We have now extended the use of these same pairs to a multiplex format in a platform called Fireplex. We also offer a full service lab for researchers who do not have access to a flow cytometer or traditionally run their multiplex assays through CROs. Our service lab is located in Boston, and our team of highly experienced scientists provide fully analyzed data and a service report for each project. The standard turnaround time for most projects is 15 business days from the time of sample receipt. In addition, our service lab team is available to customers prior to project start to help with the project design and scoping, and after completion of the project to answer any questions about the data or run additional analysis. So let me introduce you to the 3 types we are offering. As I said, we have a microRNA and protein assays, and firstly for the microRNA assay. For our Fireplex technology to work, we require the flow cytometer to have a single blue 488 nanometer laser that has detectors for green, which is for FITC, yellow for PE, and red for PerCP. As the samples are acquired on the flow cytometer and the particles pass through the flow cell as they hit the laser intercept point, the green fluorescence of the particle is used to define the particle, and different levels of green ascertain the orientation that the particle passes through the flow cell. The different levels of yellow fluorescence define the particle barcoding for a particular analyte. And using a plex file, which is defined during panel manufacturing and supplied with the panel, the software can correspond a given barcode with a specific analyte ID. The red fluorescence is used to determine the amount of analyte captured onto the particles. And these values are initially reported as mean fluorescence intensity, or MFI values. The microRNA is processed in a 96-well filter plate, into which particles and biofluids or other sample types are added directly to the well. No prior RNA isolation step is required, as an initial protein digestion step allows the microRNAs to be freed, upon which they are hybridized to the hydrogel particles. We then add universal adapters from which we perform a simple PCR. The filter plates come in handy here as the solutions in the wells can easily be eluded using a vacuum manifold, while the particles are left behind. This allows for an easy setup of PCR and post-PCR when the solution is added right back onto the same plate and particles. They are then rehybridized to the particles, and following a reporter step, the data is then read out on a flow cytometer. The data is then easily analyzed with our free software tool. Here is one example plot from our software. This is a heat map of a representative data set of various sample types run on a pre-designed FirePlex microRNA panel. The data indicates the differential microRNA expression observed across these sample types, and as you can see here nicely, the different expression patterns, and which also demonstrates that we are able to robustly detect the majority of microRNAs in this panel in each sample type. And this was done from a very small sample volume input, roughly 20 microliters of biofluids or up to 5 microns FFP tissue section curls. When comparing the FirePlex microRNA assay to the TaqMan low-density microRNA array or the Illumina TrueSeq assay, differential expression of RNAs from these 3 tissue types, including the brain, lung, and liver, comparing wells between the different platforms with Pearson values of at least 0.93 or higher, it can be observed that the Pearson values observed here between FirePlex versus TLDA and FirePlex versus Illumina TrueSeq assay correlates well across the 3 different tissue types. Here is a product snapshot from FirePlex microRNA. As mentioned previously, microRNA profiling can be performed directly from biofluids, tissue sections, and cell suspensions without doing a pre-RNA isolation step. We have a large discovery panel which can screen up to 400 microRNAs in human, rodent, or C. elegans to identify key microRNA signatures in your samples. We also have more focused panels for different disease areas that can measure up to 65 unique microRNAs in a given well. There is also the custom panel option to use sequences of microRNAs that the customer may have already validated, or mixing and matching from our predefined panels to create a custom panel. We also have on our website access to the FirePlex Discovery Engine, where you can search for specific microRNAs of interest or research for biomarkers in a specific research area of interest or in a given species. And this tool provides access to a PubMed curated word clouds where a list of relevant publications are pulled back from the search. The FirePlex Discovery Engine also accesses Mirbase, the microRNA database for published microRNA sequences and annotation. Thus, the FirePlex Discovery Engine is a powerful tool aiding customers with their microRNA biomarker research. Now, I would like to introduce you to our protein biomarker discovery platform with FirePlex 96. The FirePlex 96 platform enables high multiplexing capabilities in a 96-well plate format and can be run on a range of common flow cytometers. One of the key features of the FirePlex platform, as already outlined in my previous slide, the FirePlex 96 immunoassay is run on common flow cytometers, eliminating the need to purchase specialized equipment to run the assay. The FirePlex 96 immunoassay is performed in a 96-well filter plate, just like the microRNA, and it runs similarly to a standard sandwich ELISA. The FirePlex 96 platform enables quantitation of up to 70 protein analytes per sample per well, using only 12.5 microliters of biological sample input. And this simple workflow takes approximately 3 hours to run with minimal hands-on time. And it is run using a washed protocol. Capture antibodies for the analytes of interest are covalently conjugated to the Fireplex particles, and following sandwich formation and detection, the data is read out by scanning the particles on a flow cytometer and then decoded in our Fireplex analysis workbench software. Standard curves are generated from the protein standards provided in the kit for interpolation to allow absolute analyte quantitation. And this format delivers an assay with a superior sensitivity, sub-picogram per mil, and broad 5-log dynamic range. A key feature of the platform is the rigorous testing that is performed for each antibody pair. Each antibody pair utilized in the Fireplex platform undergoes extensive screening and validation prior to use in our multiplexing platform. In initial pair screening, we evaluate the performance of individual antibody pairs across a series of performance criteria, including sensitivity and dynamic range, as well as biological performance in native and spiked samples. We also evaluate protein interference and cross-reactivity as well, to ensure that quantitation of analytes in native biological samples matches international industry standards provided by the NIBSC and WHO. Once pair screening is complete, antibody pairs are combined together and assessed in either custom or pre-designed multiplex panel formats. As part of the pair optimization process, we calibrate the immunogen or commercial protein to biologically active protein, and the interpolation should benchmark to literature or competitor data. We also optimize the pair to avoid a plateau at the top of the curve so that the assay is within the linear part of the curve. One of the key parts to any multiplex assay is ensuring minimal cross-reactivity. The data shown here represents a pool of 26 detector antibodies incubated with capture antibodies for each of the analytes indicated in the legend. As we see, a signal is only observed when the correct capture and detector antibody pairs form the sandwich, and no cross-reactivity with other detector antibodies in the pool is observed. Here is the Fireplex 96 immunoassay product snapshot. We have 3 product offerings: discovery panels for early stage studies with 70-plex cytokine panels for both human and mouse. And we also have a more focused mid-plex pre-designed panels for specific cytokines and inflammatory markers of interest. There is also the custom panel option for researchers who have previously defined their protein analytes of interest, where you can pick from any of the 800 antibody pairs in the Abcam portfolio. Alternatively, customers can mix and match from our predefined panels to create a custom panel. As already mentioned, the Fireplex 96 platform enables quantitation of up to 70 protein analytes per sample per well. Here is our published performance data from our human cytokine discovery panel, demonstrating that this is a highly specific and sensitive sub-picogram per mil assay with no cross-reactivity that uses a simple workflow and requires minimal hands-on time. There are a number of key benefits of using the Fireplex 96 immunoassay platform. The simple workflow, low sample input, and washed assay format enables a high level of sensitivity, which is sub-picogram per mil and a 5-log dynamic range. The Fireplex assay has been designed to be compatible with existing flow cytometers, so there is no requirement for purchasing new equipment. This means it is easy to run, access, and run. The high multiplexing capabilities makes it easy to run large sample numbers in a multiplex format to get as much data as possible on which to base decisions on the drug discovery pipeline. The most recent addition to our immunoassay portfolio is the Fireplex 384. It is a no-wash, multiplexed high-throughput workflow that drastically reduces hands-on time. This format is run on a range of common high-content images. The Fireplex 384 immunoassay offers a homogeneous, 2-step, no-wash assay workflow that is fully automatable, meaning that the addition of the Fireplex particle mix assay reagents and samples can all be carried out using standard in-house liquid handlers. This simple protocol means it is ideal for larger numbers of sample wells, as the hands-on time to run the assay is significantly reduced compared to other wash-based formats. And data acquisition on high-content images takes around 10 to 20 minutes per plate. The assay input is only 6.25 microliters of biofluids and has been validated for plasma, serum, and cell culture supernatant samples. Fireplex products are supplied as ready-to-use kits that include all of the necessary reagents and labware required to run a full 384 well plate of the assay. This shows a representation of the image of the Fireplex particles from a high-content imager. The TIFF image is generated using FITSI for green to define the particle, Sci3 for yellow to define the specific particle barcode, and PE Sci5 for red to determine the amount of analyte captured onto the particles. In terms of the analytes that are available, Fireplex 384 products are available either as pre-designed panels or as a mix and match configurable panels. The pre-designed cytokine panels are available off the shelf and include options for human and mouse targets, as well as panels for organ toxicity and assessment. For a more customized panel, there is the option to select from up to 10 analytes from a pool of 50 targets that are pre-validated to work together in with and no cross-reactivity. A second pool of 50 analytes is going to be launched in the coming weeks, further expanding the offering. Here is our published performance data from our website with a typical curve using the human cytokine 10-plex panel run on a high-content imager in a no-wash format. It demonstrates that the assay gives sensitivities in the picogram per mil range by using a protocol that requires minimal hands-on time and is compatible with automation workflows. So in summary, our Fireplex microRNA assays allow high multiplexing capabilities for microRNA biomarker discovery research and no need for a prior RNA isolation step, and the readout is read on a standard flow cytometer. We also have our protein biomarker discovery platform, Fireplex 96, which is a simple washed assay format with a high sensitivity, sub-picogram per mil, and large dynamic range, also with the readout on a standard flow cytometer. As well as our more established 96-well high-plex flow-based assays, we also have our screening platform, Fireplex 384, with a simple no-wash workflow and good sensitivities. Therefore, our Fireplex platform, with the application of our highly validated recombinant rabbit monoclonal antibodies, we have developed a robust multiplexing tool offering. Thank you for listening, and we'll be happy to answer any questions you have now. Thank you.